All right, hello and good afternoon, everyone. My name is Anas, and thank you for joining us for this Only Cells webinar on antibody ID. So this webinar is part 3 and the final part of a series on antibody ID. In part 1, we looked at the basic principles of antibody identification and some of the key testing tools available. Part 2 was more interactive, where Helen went through some cases and some real-life panels. And I won't give you too many spoilers about part 3, and we'll leave that to Helen to give you an introduction. And for those of you who don't know Helen, let me tell you a little bit about her as well. So Helen Thom is a registered biomedical scientist who began her career at NHSBT as a placement student. She then secured a trainee biomedical scientist role in the RCI lab, specializing in transfusion science. And after working for many years on the bench, she then moved into the PBM team, that's the patient blood management team, where she worked on educational initiatives, and this was then followed by a project management role in hospital customer services. Helen is now part of the Transfusion 2024 program, helping develop collaborative models between NHSBT and transfusion laboratories. She's also a member of the IBMS Specialist Advisory Panel for Transfusion Science and is a strong advocate for the role of biomedical scientists. She's an excellent educator, as you would have known if you've attended any of the previous webinars, and she brings a really thoughtful and engaging approach to teaching. And once again, we're very, very lucky to have her leading this series. So with that, a huge thank you to Helen for 2 amazing sessions. And now on to the final one. I'll hand it over to you to get us started.
Lovely, thanks very much. Thanks again, everyone, for attending. I hope you've enjoyed the first 2 sessions. So as Anna said, this is the final session. I'm going to crack on because I know I have a tendency to talk. I haven't included polls this time, just because there's so much information that I want to get through. But it's split into sections. So at the end of each section, I'll stop, and if there's any questions, I'll take them, and hopefully there'll be some time at the end. So today I'm just going to cover a brief recap of some of the tools we've discussed in previous sessions. This is going to be a very brief overview of high incidence antibodies. We're going to cover some advanced techniques, so DTT treatment, neutralization, inhibition, and alloabsorption. And then I've just got a couple of cases to go through, just by way of an example. This is a really good, hopefully it'll be a really good talk, but each one of these could easily be an hour on their own. So it's a very much a whistle-stop tour. So we've discussed in the last couple of sessions about the basic principles of antibody ID. So you've essentially, for your antibody ID, you're mixing unknown antibody with known antigen, or if you're doing grouping and phenotyping, you're mixing a known antibody with your unknown antigen. In either of these scenarios, the antibody will bind with its corresponding antigen, and that is the basic principle of antibody ID, and you'll see underpins everything that we're doing. We've talked about grading and how this can be really helpful when you're doing your antibody investigations, sort of particularly if there's mixtures of antibodies or is something enhanced, by enzyme, for example. Speaking of enzymes, so we've discussed a lot around papain treatment. You'll be aware that I'm a little SpongeBob SquarePants fan. So we'll have Patrick here, who is papain, and when you have antigens which are sensitive to papain, it's essentially just like chopping the trees down, and it leaves behind different antigens. So some antigens are actually enhanced, so you get really strong reactions. So the Duffies and the M, N, and S, they can be destroyed by papain, but it'll enhance your sort of RH and Kel, and the kid is left behind an enzyme as well. Talked a lot about knowing the clinical significance, because this is depending on whether your antibody is clinically significant or not, is going to sort of dictate your blood selection for these patients, which we can see today can be quite difficult. We've talked about your positive DAT and scenarios when you would perform a DAT. This can feed into when you would perform an elution. And today you'll see it sort of determines whether we're going to perform an alloabsorption as well. These are just some of the results that you can get when you do your DAT. So it might be IgG pos, could be C3d only, or a mixture of both. You can even do monospecific DATs, which will give you even more, such as IgM. So moving into today's session, so we're going to talk about pan-reactivity. So I've intentionally put this because it's sometimes it's apparent pan-reactivity. And when we're talking about pan-reactive, it comes from the Latin word meaning all, every, whole, and all-inclusive, which I thought was quite sort of apt. So it's basically you can get reactivity with all cells. With high incidence antibodies, these most often appear as pan-reactivity, but with a negative autocontrol or negative DAT. But I've put that this isn't always the case, because again, sometimes it can just depend on the patient themselves and whether they've been transfused or not as well. The antibody will bind to its corresponding antigen. So in your high incidence antibodies, when that antibody is binding its corresponding antigen, it happens to be with over 90% or even over 99% of your donor red cells, because these are from humans and the donors are 99% will have the antigen, which is what makes these antibodies sort of high incidence. You're making an antibody against something which the majority of the population are positive for. The patient's autocontrol is negative because their own cells are antigen negative. So there's nothing for the antibody to bind to, and this is why you tend to get that negative autocontrol. But like I said, it's not always the case. Pan-reactive antibodies, however, most often appear as pan-reactivity with a positive autocontrol and or DAT. And this can be IgG, IgM, complement, or a mixture of all of them. The patient's autoantibody will bind with all red cells, and it includes their own. And when we've talked about autocontrol, when it is positive, there's IgG antibody present, or antibody present anyway. And that's why you get this sort of pan-reactivity. So either one of those looks pan-reactive, but we do consider the autocontrol. So I've just got a panel here. So when you're looking at high incidence, and I put what you typically would see. So what you would get is it could be pan-reactive by enzyme and IAT. You have a negative autocontrol, and I've just included a reagent control on the bottom there, just in case anybody was wondering what that was. So you're getting this pan-reactivity, but with a negative autocontrol. But that's what you'll typically see. You don't always see. It's going to depend on the antibody and how the antibody behaves. But what you can't see is if there are any underlying alloantibodies. Or this could be an alloantibody as well, because it could be a high incidence alloantibody, but you don't know if there's mixtures of things going on. It could even look like this. So this is just another example where you may have something going on by IAT, but this particular high incidence antibody is sensitive to papain, so your enzymes could be negative, but you have that auto control is negative. And again, we've just got reagent control there. And this is very much just going to depend on the kind of antibody that you're looking at. But again, we can't see if there's underlying alloantibodies, and we haven't identified what this antibody is yet. But these are just things that would make us suspect something high incidence. So I've put here some common examples. I'm going to just very briefly talk about these because these are what you may have heard of in your lab. There'll be thousands of these. But again, I don't have time to go through them, but I will point you to some resources at the end. So in your M&S system, you've got an EU, which is a high incidence antibody there, and you need to have antigen negative blood. You've hopefully heard of anti-Duffy3. Again, you need antigen negative blood. You've got anti-little k, which is your Chelano, again, antigen negative blood, and it's depending on what the antibody is as to whether you need antigen negative blood, and that goes down to the clinical significance of the antibody that you've found. We've got a GK3 that you may have heard of, again, antigen negative blood required. You've probably heard of Chido Rogers. They are two separate things, but they look very, very similar. And when you're selecting blood for these, if you went in your BSH guidelines, it's looking at sort of least serologically incompatible. And it's a similar picture with your Knopps McCoy antibodies. So I did say it was going to be very high, quick overview. So one of the things that we're sort of looking at when you think you've got a high incidence antibody is that apparent pan-reactivity with the negative auto. But as I said, it's not always. The patient's ethnicity can indicate which antibody you may be looking at. So in the Black population, if your patient is FYE neg, FYB neg, they may have a mutation which would allow them to make an anti-Duffy3. In the Caucasian population, 0.2% of the population are Chelano negative, so you could make an anti-Chelano. The rare cell panels can be performed. These are really similar your normal antibody panel that you would see in the lab, but they are negative for any high incidence antibodies, but they do include extended phenotype. So they'll, if it was UNEG, the cell would still also be GK, POS, GKB, neg, that kind of thing. So you can actually get your exclusions for your other antibodies. Might need referral to IBGRL as well. And they have lots and lots of weird and wonderful things they can do, even more weird and wonderful than RCI. We've got a lot of serological clues, so it's how the antibody behaves, whether it's papillon sensitive or is it enhanced. We can use grading. This is where those tools come in, because if there's, if it's a sort of weak antibody, you might be thinking it's Chido. If it's negative by enzymes, that would indicate Chido. Or if it's enhanced in enzymes, you could be thinking Knopps McCoy or something. And your patient's phenotype is a really useful tool when you are looking at high incidence antibodies, because if the patient was big S neg, little S neg, for example, that would make you think that you're looking at an anti-U. So lots of different clues you can use. When you're selecting blood for these patients, the high frequency antigen, because over 90% of the population are going to be positive for these antigens, it is difficult obtaining blood for these patients. You might even need to go to the frozen blood bank. Sometimes you've got multiple antibodies as well, so not only have they got an antibody to a high incidence antigen, but they also have other antibodies, so it just makes it even harder to find blood. And depending on the significance, some require antigen negative blood, which can be really challenging to find, but those ones where it's the least serologically incompatible, basically just put up your few and sort of hope for the best. So I'm going to take you through a case here. So this is a real case that I worked on when I was in the reference lab. It's a few years old now, but I just thought this is a good way to show some of the work that we had done in understanding these antibodies. So we've got a patient, it's an 85-year-old male. We've got a request through for an antibody ID in 2 units of blood. They've got anemia, hemoglobin of 70. They were transfused 2 months previously. And the current findings from the laboratory who referred it, where they've got historically their OPOS and their known big C neg. This time they've got a POS DET and the antibody screen was positive. So this was the first investigation. So I'm just going to walk you through this. So we've found an anti-big C. It's quite happy that that was there. This one, it looked a bit like an anti-little e. And the reason I'm saying this is line 5 is actually a heterozygous line when you're looking at your little e. So I was like, and you just kind of get a feel for what things look like with your panels when you're doing these all the time. But with a little e, you would expect line 3 to be negative, but in this case it wasn't. So I was just a bit like, this could be this. What we also have as well, using the grading, is that there are some, this one, it looked a little bit, I put GKE-ish, because that was what I would actually do. And I'm like, oh, you can almost like sniff them out and you think, right, I think there might be a GKE here. But you would need to do another panel to sort of confirm different things. So, and as well, this patient has a positive autocontrol, so it's going to require a DET. So I put another panel up. This is our panel 2, so different to the primary panel. Again, the big C was there. This time, I'm like, yeah, that definitely looks like an anti-little e. And even line 3 is negative on this one. And I was still convinced, just looking at the strength of reactions, that there was a GK poking out of the top of it. So what we have so far is an anti-big C, an anti-little e, possible anti-GK, but I didn't know if there were allo autoantibodies or the clinically significant. That's going to depend whether they're allo autos. What is the phenotype? Can we phenotype? This patient was only transfused 9 weeks earlier. And if you've listened to us in the previous ones, we're always saying don't perform phenotyping if the patient's been transfused within the last 3 months. It's got a positive autocontrol, so it needs a DET. The patient hasn't been transfused within the last 6 weeks, so technically you wouldn't be doing an eluate on this, but they have made antibodies. So in this case, I've just gone against everything that I've told you for the last 2 sessions, and I went with my gut. And I did an eluate, even though it was outside of the time. And what I found was they had a pan-reactive antibody in the eluate. And it was 3+ uniform reactions. So this is typical of the sort of that pan-reactive. It doesn't look like there's a mixture of antibodies going on in the eluate. And we're a little bit naughty and we did phenotype this patient. So there was no dual population confirmed, sort of the patient was big C negative, and we did do some kid phenotypes as well. But we did the JK and the JKB. So again, going against everything that I've said to you recently, but it's what we did. So we said we've definitely got an allo anti-big C. We could confirm the presence of it. The patient's little e positive, so this is looking like an auto little e. These are unlikely to be clinically significant. And there was a JK poking out by enzymes, but we couldn't confirm due to the presence of the auto little e. And the little e neg panel that RCI use isn't papanized, so even if we'd use that, it's by IET only, so we couldn't use it to confirm it. But yeah, like I said, you can sniff kid antibodies out, and if you get a feeling, just regard it as an allo antibody and think of it as clinically significant if you suspect there's something there. The DAT was 1+, so I'd done the earlier, it was 3+ pan-reactive, so I issued 2 units of O-pos, big C neg, GK neg. So saw the patient again a month later, so we've got that historical phenotype on file. The anti-big C was there, the JK was there this time, so we're able to confirm that, and this time it was a pan-reactive enzyme antibody. That had gone to 2+, so it was 1+ the first time we saw them, this time it was 2+, so that the strength had increased, which prompted us to perform an eluate. Again, it was pan-reactive. So we issued 2 units of O-pos, big C neg, JK neg, and we gave them an updated antibody card because we confirmed that the presence of the anti-JK this time. Saw them again a month later. This time there was no change in the serology, so the big C was there and the JK. We didn't do an eluate because there was no strength in the DAT and no change to the serology, so I issued the same blood again. And then we saw them in October, and I remember this because I was going into on-call, so it was on a late shift. It's always on call when you get something challenging. So initially it didn't look challenging, so I was like, brilliant, we've got that enzyme 1 anti-big C, that's starting to sort of dissipate now, it's not there by IUT anymore. There's a previously reported allo anti-JK because kid antibodies, they can be there one time and then they disappear, and this one's disappeared, so it's lucky we kind of caught it in the beginning poking out, because we may have issued antigen positive blood. However, the DAT has gone from 2+ to 3+ this time, so it does require an eluate because there's been a change in strength in DAT and the patient has been recently transfused. So I did an eluate. I won't tell you what I said when I worked out what it was, but I went, oh, that really looks like an anti-little k. My patient is big K positive. Uh-oh. That was probably the nicest way that I can put that. There may have been some expletives going along. So we're looking like we've got an anti-little k in the eluate. So remember I'd said last time when we talked about inclusions and exclusions, if you find an antibody, you've got to have 2 positive examples and 2 negative examples to confirm the presence. And it's really important that you exclude the presence of any further clinically significant antibodies. So we don't want our Squidward to have a transfusion reaction. So I put up another panel because I needed more cells to confirm the presence of the little k. So again, this is in the eluate, it wasn't coming through in the plasma. And it's really looking like a Chilano. So I was like, uh-oh. I also put some rare cells up. So we have our, well, not quite rare cells, but R1, R1 panel and R2, R2 panel. Because I wanted to confirm that the anti-Chilano was there. These happen to be on the cells I'm confirming happen to be antigen negative for the other antibodies, even though they weren't coming through by IAT at that time and they weren't in the eluate, but just to cross the T's and dot the I's, as it were. So we'll go back to phenotyping now. So the Chilano phenotyping is anti-IG, it's IgG anti-sera. So we know the patient is K-pos, big K-pos, sorry, but other little K-pos, we don't know. Only 0.2% of the Caucasian population are little K-pos, so I had a 9% chance that this was going to be heterozygote. I was like, please be heterozygote. It's not looking like it was going to be though, given that they've got the antibody. So if the patient hadn't been transfused, they have got a positive DAT. So if I did Chilano phenotyping, I would get a false positive, so I wouldn't be able to use that. But they have actually been transfused, so I wouldn't be able to do phenotyping anyway. So I can't do phenotyping for this patient. However, we can do genotyping. So remember the last time I was saying it's a really useful tool when a patient's been transfused and or has a positive IgG DAT. So am I going to have blood for this patient was what I was wondering. And this was actually the unit that I was cross-matching at the time. So for you, when you want to work out how many units of blood you would have to randomly select in order to have one there. So have you got it on the shelf effectively? You want to be just, you can calculate your frequencies. So the patient in this case, the patient's C neg, that's around 32% of red cells are going to be negative for the big C antigen. Around 24% are going to be negative for the GK antigen. And to be Chilano neg, there's 0.2% of the population. So if I was to just go into the fridge and randomly select units without knowing what the ABO was, I would have to test around 22,000 units to get these 2 units. So it's pretty scary. We know that the group O, so if I chose red cells that were group O, I'd have to test 13,000. So that's a lot of blood. I was really hoping that the unit that I had was going to be Chilano negative. So NHSBT has a system called Pulse. So any blood donation that is ever taken goes into this system. So I could look on the system to see if this unit was Chilano negative. And you can see there's where I've circled, it's K positive, and where it's got a plus sign, that testing's been performed twice and confirmed. So we know this unit's K pos. When it has a little p, it means it's been typed once, but it's Chilano positive. So this unit is heterozygous, which would fit because that's 9% of your population. So what I did was you can search. So the really good thing is I was up in Newcastle. I didn't have any units in Newcastle, but there were 4 units that matched this phenotype in around different places in the country. So what I did was I searched for them, I reserved them, and then we got our transport department would send those up. So you work closely with hospital services and transport, get them sent up. So what we had was the results for this patient. So thankfully I didn't have to type 13,000 units myself. The testing department had already done that for me. So we've got our enzyme, only allo big C, previously reported GK, in a Chilano and a pan-reactive enzyme in the eluate. So I've got the blood sent up, and that was O pos, was what was just group O, because I don't think there was any O pos. It was O negs we ended up issuing. Big C neg, JK neg, Chilano neg, they were ordered and issued the following day. So it didn't actually take that long, which was really good. And we gave an updated antibody card. So we couldn't perform the phenotyping due to the IgG and the recent transfusion. Genotyping was performed on the subsequent sample and we confirmed the patient was Chilano negative. So that was just to show you what those cases can be like. That was a real case and it was quite hairy at some points, but thankfully it wasn't urgent. Obviously that's when it can make a difference, and the patient had the blood, and after that we just used to order them on a regular basis for them. So you can see there everything that I've talked about to find the testing those 13,000 units to get the blood that we needed for this patient. We've looked at the patient history, with the hospital had done a screen, we've looked at grading, we've done antibody IDs, we've talked about inclusions, exclusions, we've used our panels, we've got our panel sheets, understanding the clinical significance, we've done phenotyping and genotyping, yeah, DAT and elution. So everything we've covered in the last couple of sessions shows how, even though this is a sort of high incidence antibody, you're using the same techniques to go through. So I'm going to move on to modification now, unless anyone quickly has any questions around that case before I go on. Can't see anything, so I'm going to just crack on. Thank you. So now we're talking about modification. So as well as using the rare cells to identify your high incidence antibodies, you can actually modify either your red cells, so your reagent red cells, or your plasma. And this can help you to identify certain antibodies. So I'm going to talk in a minute about DTT, but we've also got neutralization, inhibition, and alloabsorption. So DTT, so this is a chemical, I don't know if any of you have used it in your lab, but it's very smelly. is what I would say. So it's a reducing reagent that breaks disulfide bridges. So it's really helpful when we're in the blood bank, particularly for patients who are on anti-CD38 therapy, so say daratumumab, for example. So when the reagent red cells are treated with DTT, it disrupts the antigen structure because it's a disulfide. It reduces your disulfide bridges. The antigen, the CD38 antigen, is on all red cells. The patient's on monoclonal antibody therapy, which targets the CD38 antigen on the red cells, which is great for treating patients with multiple myeloma, but it's not great when you're trying to do an antibody ID. What we need to be mindful of, though, is when we use DTT, some antigens are sensitive to it, such as the KEL system. So the KEL system antigen is essentially that it's the KEL cloud, and it's held together with disulfide bridges. So when we treat them with DTT, it removes those antigens from the cell, so you can't exclude them when you're doing your antibody ID. So I've tried to just do a sort of very basic overview of this. So this is just your normal antibody ID. The screen, if we go back to session 1, you've got your patient's unknown plasma with the known red cells. You mix them together, add a bit of VHG, and you get agglutination. And this is what it looks like. So you've got agglutination in those first 2 cells. And this is the basic underlying principle for what antibody ID. So when we are looking at our red cells, they have the CD38 marker on them. All red cells do, including our panel cells. So when you do a normal panel, it's untreated. What you're getting is the antibodies, the anti-CD38 antibodies will bind to their corresponding antigen, which is the CD38 marker, which is present on all red cells. So you get a pan-reactive either screen or panel. So that's how it works. So it's interference. It's not a clinically significant antibody, but it prevents work from seeing if there are any underlying alloantibodies. So what you would typically see, and again, it's typical, it may not exactly look like this, but you have your pan-reactivity. So there we've got pan-reactivity in IAT and enzyme. And negative autocontrol, it may be positive, it depends again on the patient. I've seen a mixture of both with your daratumumab patients. So you've got this pan-reactive antibody, but you can't see if there's anything underneath. So what we do is, in this case, we're going to treat the red cells. So you're treating your reagent red cells in order to find out if there's an underlying alloantibody. So what we're going to do is we'll add DTT. This disrupts those disulfide bridges on the CD38 marker. So it removes them from the red cell, but it also removes any DTT sensitive antigens, such as your KEL system. Then we'll do a panel. So this is my DTT panel. So if anyone in RCI is like, oh yeah, I've done a DTT panel. So we've treated the reagent red cells. They've got the same antigen profile as your normal red cells, apart from you can't exclude your KEL system antibodies because they're effectively antigen negative. So this is what you would see. So you have the negative reactions by IAT. But it allows you to see if there's anything underneath. In this case, there isn't, so it's negative. So you would see pan-reactive antibody caused by the monoclonal antibody therapy, but no underlying alloantibodies, but unable to exclude your KEL system. But it could be like this. So in this example, we've done a DTT panel, and the patient actually has an underlying Duffy B, but we couldn't see that that was there because that was previously masked by the anti-CD38 pan-reactivity. So like I said, you can just see why it's a bit of a pain. So when you're selecting blood for your DARA patients, if you'd want to be matching in accordance with the BSH guidelines. So ABID and K compatible and extended RH matched. You can perform full phenotypes and genotypes for these patients, but you don't need to give antigen negative blood. But you do need to give antigen negative blood if they've made a clinically significant antibody. So in that previous example, we'd be given ABID and K compatible, extended RH matched, and Duffy B negative. And also any special requirements that the patient may have. But again, this is going to be patient specific. So now I'm going to move on to neutralization and inhibition. So this is a sort of useful tool, and I've called these nuisance antibodies because they're just a bit of a pain. And what we'll do this time, so in the previous example, we've treated the red cells. This time we're going to treat the plasma. So you can either treat your patient's serum or plasma. You pre-incubate it before you do your antibody ID. So you can pre-incubate it with a reagent. Depending on the antibody, it's going to depend what reagent that is. And then you just do your investigation in the normal way. So Chido-Rogers, the sort of classic one, I think that everybody would know. So Chido-Rogers antibodies will bind C4 complement proteins. And what you can do is you can neutralize the antibody by adding excess complement. So you basically just flood the plasma with complement by adding AB serum, and it stops the antibodies from binding to the red cell. We also have Knopps-McCoy antibodies. So this one binds complement receptor 1. The antibodies are inhibited, and there's reagents you may have heard of called soluble recombinant proteins, which are basically just manufactured proteins that you can add to the plasma to block the antibody from binding the red cells. So I'm going to talk you through how those work now, because to me, everything's better when you use pictures. And these soluble recombinant proteins can also be used for monoclonal antibody therapy. So when you see anti-CD38, you can either treat the red cells or you could treat the plasma. When you're doing the soluble recombinant protein, so I've already mentioned that anti-CD38 binds the CD38 marker on the red cells. Your antibodies this time, you can inhibit them by adding the protein, which is SR-CD38. This has got no reactivity with clinically significant red cell antibodies. So what you could do, instead of using DTT and not being able to exclude your cell system antibodies, you can just treat the plasma instead, and then you're actually able to exclude your cell system antibodies or other sensitive DTT sensitive antibodies. So I'm just going to use the C4. So this one would be your Chido or Chido-Rogers. So you've got the C4 complement protein on your red cells. So what's happening this time? You've got your patient sample. They've got Chido-Rogers antibodies in their plasma. When you mix it with those reagent red cells, which have the C4 complement protein on them, you're going to get pan-reactivity because the antibody is binding its antigen. So your Chido-Rogers antibody is binding the C4 complement protein antigen on your red cells, and you get pan-reactivity. And again, I've put this as what you can typically see. So remember I was saying at the beginning, it really some of the clues that we're looking for is how the antibody behaves. And Chido-Rogers antibodies are papain sensitive. So if you had something pan-reactive, so we've got a 2+ pan-reactive by IAT, it's negative in enzymes and the autocontrol is negative. That would be a little clue in the lab when we were doing the antibody ID to say, oh, I think this is a Chido-Rogers antibody. Then you could go on to use your rare cells to sort of get further exclusions and confirm, or you can neutralize the antibody if you know what the antibody is and you use the correct reagent and neutralize it. So that's what we do. So said there, you've got a pan-reactive antibody by IAT, but you're unable to see if there's anything underlying. So what we do this time, take your patient plasma and we add AB serum. You just mix them together. I think it's a 2:1 ratio, and you're adding this excess of complement. So you're neutralizing the antibody because you're basically blocking it from actually being able to bind to the red cells. So we'll add it in excess so that it blocks it. And this is what it looks like. So it's very busy on this slide. I do apologize, but just wanted to show what it was like when you were doing your screen. So you've got your Chido-Rogers antibodies, your C4 complement protein on the red cells, that excess of C4 from the AB serum. So the antibody can't bind. So when you do your antibody ID, you've neutralized that antibody, so you get a negative result. And then when you get your panel, this is what you would see. So you've got your IAT there, your 2+, you've got your AB serum, and that is negative. So that's neutralized those Chido-Rogers antibodies. There's nothing underlying. What you will see in the next column is I've got PBS, and that is a saline control. So when you are neutralizing your antibody, it's really important we're diluting the antibody down. So what you need to show is that you've neutralized the antibody, you just haven't, not that you've diluted the antibody out. So you use a PBS control. So you would dilute the patient's plasma in the same ratio as you would with the AB serum. You test both of them doing your antibody ID with your panel, and that's the original enzyme, which is negative. So what we've proved here is we've neutralized this antibody with AB serum. Then you would go on to confirm that this is Chido Rogers. Either you could perform phenotyping, probably done at your reference lab, I would imagine, and using those rare cells to confirm. So you'd have like your positive and negative examples as well. So it's a similar picture for your soluble recombinant proteins. So I've grouped the two together just for time here so I can get to talk about everything else. So this is either your CD38 or your Knopps McCoy. You've got your soluble recombinant protein, so you're adding that to the patient's plasma. Again, it's in excess, so it's inhibiting the antibody. And this is kind of what you would say. I've used Knopps as an example here. And again, this is shown how the antibody behaves. So it's by IET and enzyme. So that's kind of a clue as to why you might be thinking that it could be this sort of antibody. You can use your rare cell panel as well. And you've got your auto is negative there. And again, it's always going back to, it's just saying you can't see if there's anything underlying. Sometimes you may have something like if there was an underlying antibody that was really strong, you might have different strengths of reactions. And even within the panel, this is, I've made these for this talk, but they are made from human red cells, like your donor red cells. So the number of antigens on the actual cells could vary. So you may actually get different grades of reaction as well. And it's enhanced in enzymes. So again, that understanding how the antibody behaves. So this is what we typically see. So you've got your IET and your enzymes are positive. Using that soluble recombinant protein, you've inhibited them, and you also need to run the PBS control as well, just to show that you haven't diluted the antibody out. So here we've neutralized it, PBS control, and there's nothing underlying. So before I move on to alloabsorption, has anyone got any questions?
We've got a question in the Q&A, and the person's asked, can you use DDT outside of monoclonal antibody testing? For example, you present with an anti-little k, remove this LANA to see what's left, or does it remove lots of others, not just the KEL system?
That's a really good question. And yes, you could use that to remove Chelano if you wanted to, because it does denature KEL system. There's other antibodies that it does neutralize as well. So Knopps is actually neutralized, denatured when you use the DTT panel. So if you had a Knopps antibody, you could actually perform DTT treatment of your red cells as well, would be another way to use that. So that's a fab question, and yes is the answer to that. So you can use it outside of it. It's more commonly used in the monoclonal antibody therapy, but yes, you could absolutely use it. So fantastic question. I think I might have to steal that one. Thank you. Thanks, Emma. Right, so that was a really good question. I'm definitely pinching that. So alloabsorption. So we've got just under 20 minutes left. So this is going to be the quickest tour of absorption ever. But now is time to introduce you to SpongeBob alloabsorption pants. It's one of my favourite sort of ways to explain how you do alloabsorption. So this is going back to whenever we've talked about DATs, we're saying you've got to sort of consider the patient's history, the clinical information, and results of any other lab tests. So alloabsorption is typically done when the patients have got autoimmune hemolytic anemia. There could be other conditions, but this is the sort of classic one that you would see. You've got this pan-reactive antibody and a positive auto control. The DAT is IgG positive with or without C3d. So these are the things, if you've got all this combination, we would be thinking, right, we're going to have to do an alloabsorption here. So one of the things you need to think about is, again, it goes back to that understanding how the antibody antigens interact with one another. So we'll have SpongeBob alloabsorption pants. You've got to think about your papain, and it's just how the antibody binds with its corresponding antigen. So when you have the pan-reactive picture and it's something like this, not a high incidence antibody, just an actual pan-reactive one, you've got those pan-reactivity, generally really strong reactions enhanced in enzyme, and a positive auto control and positive IgG DAT. And then just for good housekeeping, we've done a reagent control as well. But we don't know if there's any underlying antibodies. So how does alloabsorption work? So if you think if SpongeBob is an alloabsorption cell, so he's a red cell that's made, he is D positive, so he's really good. If you had an anti-D, he could remove anti-D because the D positive on SpongeBob would remove any anti-D in your patient plasma. SpongeBob is also been papain treated, so he's really, really strong. So you got those. So SpongeBob is great at getting a hold of your RH antibodies, really strong, but also really good at getting a hold of alloantibodies as well. So if you think about your antibody-antigen interaction and the antibody will bind its corresponding antigen, SpongeBob is D positive, so he's going to grab a hold of the anti-D, so you can see the black Y's there are your anti-D, so he's going to grab a hold of them. So when you mix the SpongeBob with your plasma, he's going to remove those the anti-D, but he's also really good at getting a hold of the autoantibodies, because remember, autoantibodies will bind with all antigens. So the autoantibodies like, come on SpongeBob, grab a hold of me as well. So what he does is he grabs a hold of them. However, he can't remove anything that's sensitive to papain, because the Duffy, for example, the Duffy antigen has been denatured because SpongeBob has been treated with papain, so he can't remove it. So it's knowing what's removed and what's left behind, and it can get complicated. So I hope I'll sort of keep you on board. So I'll just show you. So one of the things when we're using alloabsorption cells, we know what the profile is, because you need to know what you've removed from the plasma. So if you look at the profile here, you've got your CD, E's, your Kel, and your kids, but the effectively antigen negative for your MNSs and your Duffies, because they're papain sensitive, so they've been removed. You might have noticed the eagle-eyed amongst you that both the cells are big E negative and they're both little e positive. So it's really handy to know what phenotype, but there is an R2R2 alloabsorption cell available, but the typical ones we tend to use are the R1R1 and little r. But if the patient had a little e, we could use the R2R2 absorption cell to see if there was anything sort of underlying. Hopefully this will just become clear as we go along. So how do we do the absorption? So you've got your patient's neat plasma, and we've got 2 lots of SpongeBob. One is an R1R1, so that's a D positive cell, and a D negative cell. So you take a ml out of the neat plasma and you put it into the R1R1 cell. Then you take another ml separately from your patient's plasma and put it into your little r cell. Give them a really good mix, and you incubate them at 10 minutes at 37 degrees. You can add a cold step if the patient's got a cold antibody as well. Once they've incubated, you centrifuge them and you take of absorbed plasma, and then you repeat the process, because we want to be making sure we do this twice to make sure that we remove the antibodies. But because what we want to do here is get rid of that autoantibody. If you've got really strong examples, sometimes when we perform additional absorptions as well. So the antigen positive absorption cells remove the corresponding antibody from the patient's plasma as well as the autoantibody. So this is what you end up with. So you have your neat plasma, you haven't absorbed this, and it contains a pan-reactive auto because you've already done that part, but you don't know if there's any alloantibodies there. Then you've got your R1R1 plasma. So for this one, you've absorbed out your pan-reactive auto, plus anti-D and big C if they're there, and little e if they were all there, plus anything else depending on the antibody profile. So in this case, you could remove JKE. So this could contain antibodies. We've taken some out, but there could still be some left behind. And then with the little r, you've absorbed out anti-little c and anti-little e if they were there, plus whatever else depending on the profile. So this case, you could remove big K and JKB if they were there. And this could contain antibodies. So when you do your antibody IDs, I've got here, this is the sort of what you would be looking at when we've done the absorption, we've got our neat plasma and what R1R1 absorbed plasma and the little r absorbed plasma. So the R1R1 plasma, so you can see here, the neat is 4+ in strength, and then I've got negative reactions against me, R1R1 and the little r. So the R1R1 plasma is removed the pan-reactive autoantibody and there's nothing left behind. The little r has also removed the pan-reactive antibody and there's nothing left behind. So here we have a pan-reactive autoantibody, but there are no underlying alloantibodies detected in the alloabsorbed plasma. So we're mindful every time we know the profile of these, we know what it will remove, and you test them in pairs so that one is always going to be antigen positive and one is always going to be antigen negative, or some could both be antigen negative if it's, say, Duffy's, for example. So it's knowing what's removed and what's left behind. So when you're selecting blood for your patients with autoimmune hemolytic anemia, you want to do ABO, extended RH, and K, and this is to prevent alloimmunization of these patients. It needs to be antigen negative for any alloantibodies that the patient has. It's issued as suitable because your neat cross-match is actually, it's technically incompatible because you'd get a positive cross-match, but we've modified it so we know that it's okay for the patient, so it's suitable. But what you need to be mindful of is you may have absorbed out a high incidence antibody. So this is why, you know, it's all these things that you've got to consider when you're doing these investigations. And this is just an example of what your cross-match might look like. So you could have a really strongly incompatible with the neat plasma, but if you test it against the absorbed, you've taken out that pan-reactive autoantibody, there was nothing underneath, and you've got negative reactions with that. So you can even test the cross-match against the absorbed plasma as well. So I've just got a few more examples here. So this time, you can see there, it looks like we've got an anti-D. So you have your neat plasma with your pan-reactive antibody, and then it looks like it's an anti-D underlying. So if we look at the absorbed, so it's that knowing what's been removed, knowing what's left behind. So the R1R1 cell is D positive, so can remove anti-D. The little r cell removes the pan-reactive autoantibody, but it can't remove the D antigen because it's D negative, so it has to leave it behind, and there is a D left behind, so there's an anti-D there. And then just to confirm that there's an anti-D there, it's been removed from the D positive R1R1 absorption cell. So it's left behind with the D negative cell because it can't remove it, but it has removed it, so that almost just confirms that, yeah, that's there, because one, it's been left behind in one and it's been taken away with the other one. That's why it's really helpful to know the profile for these cells. So in this case, we've got a pan-reactive auto and there's an anti-D detected in the plasma, but depending on the patient's phenotype, it's going to be allo, auto, or non-spec. So another antibody ID here. So this time, we've got an anti-Duffy. So remember, these cells are patternized, so they're both Duffy negative. So the R1R1 plasma is removed the pan-reactive auto, but it can't remove Duffy, so Duffy is left behind. And it's the same picture with the little r, so it's removed the pan-reactive autoantibody, but it can't remove the Duffy because it's been patternized, so it's left behind. So you can even use it, this is how you would use it, it's knowing what's left behind. Does it get absorbed out by one if it's antigen positive, or are they both left behind, as is the case here? So this time you would get a report saying you've got a pan-reactive auto and an anti-Duffy detected, and again the phenotype is going to depend on the patient's phenotype, whether it's allo or auto, or non-specified if it's been transfused or has a positive TAT. And just looking at the last one here, so just this is a bit of a combination one. So we've got an anti-M there, and we also have a K, but you will notice the K is only present in the R1R1 cell. And this is because the R1R1 cell is K negative, so it can't remove anti-big K. So the R1R1 K neg plasma is removed the pan-reactive auto. It can't remove anti-M because it's papain sensitive, and it can't remove anti-K because it's antigen negative, so the M and the K get left behind. And with a little r, this cell's K positive, so it can remove the pan-reactive auto, can't remove the anti-M, but it can remove the K. So it is that just knowing what's removed and what's left behind. And it's using the clues from the antibody binding to its corresponding antigen, either removing it or leaving it behind as to what's in here. And just a little note, because I like it just for completeness. If it was confirming the presence of these antibodies, I do actually have 2 positive examples and 2 negative examples, but I couldn't use that cell because it's positive for both of them. So hopefully that's shown you how alloabsorption works. Are autoantibodies clinically significant? If you look in the BSH guidelines, most of them cause no clinical problems, but it's saying there clinically benign autoantibodies often cause the lab problems, which they do, because we can't really see what's going on underneath. Selection of blood may be influenced by the presence of an autoantibody or a mimicking one, so autoantibody little e's can be a pain, and depending on the ethnicity of the patient and things like that, it might be a little E variant, so it just can get a little bit complicated. But that's antibody identification for you. It can be very complicated. And this is why this session is called the weird and the wonderful. So there are other times we can use alloabsorption. So it's typically used for autoimmune hemolytic anemia, but actually it can just be used to separate out complex mixtures. And it's really good in G investigations. Now, a G could be an entire talk, so this is going to be the quickest talk of G you've probably ever had in your life. But it's just to show how you might identify anti-G. And it's really important with D that you need to distinguish between D, big C and G, because it can prevent sensitization. You can be eligible for anti-D prophylaxis if you're pregnant, and it can also assess for the risk of HDFN. So you've got a friend in G. So if you're wondering what I'm talking about and you've not heard of G before, G is best mates with the D antigen and the big C antigen. So wherever you have anti-D, wherever you have the D antigen, you have a G antigen. And wherever you have the big C antigen, you have the G antigen. So they just like to hang around together. So if you're G negative, you are D negative, big C negative, and you have to be G negative because it doesn't want to hang around with you because neither one of them's there. And I've just put here some common G-pos and G-neg phenotypes. So basically, there's not really a lot of G-neg blood kicking around there. So if you want some G-neg blood, you're looking at your little r phenotype weights, D-neg, big C-neg, G-neg. I put the other 2 at the bottom there, and I'm saying they're not really common because it's like less than 0.01 of the population or something like that. So going on to how we would use alloabsorption to help us identify G. So if you have an anti-D, your panel would look like this. You've got those sort of first 3 lines are positive, strong reactions on line 3 because you have R2 cells. This cell here is big C negative, so you get loads and loads of D antigen on there. So that's a really good clue when you've just got anti-D. That line 3 is the strongest one out of those, and it's enhanced by enzyme. When you've got your anti-big C, you can see similar picture there. You've got 3+ reactions by IAT, enhanced reactions in enzyme. If you've got anti-D plus C, it might look a bit like this. So you've got the line 3 again is the strongest reacting one out of those. But if you've got anti-G, you can't really tell if there's a D or a big C there. But there are some little clues that we're going to talk about. So this one is like a spot the difference is probably the best way to do it. So on the top there, we've got a D plus a big C. And on the bottom, is it D plus C or is it G? So there's a few clues as to whether it might be. And this line 3 is the key. So R2, so line 3, has more D antigen than R1. So it's steric hindrance. So because the big C is not there, you can just fit on a lot more of the D antigen. So in D plus C, you would expect to see a stronger reaction versus that R2 cell. In G, you get a weaker reaction versus R2, and it can indicate G. It doesn't say it's there, but it can't exclude D or big C. But if that's sort of weaker than your other ones, you might be thinking, hang on, this could be a G. And the other clues in the reference lab, but it can get really confusing, but I'll point you to a talk later on. And the R1, R1 are stronger because you've got D and C, so effectively you've got a double dose of G. So it's the same procedure when you're performing your alloabsorptions, but the panel profiles are different. So here we've got an RO little r and an R dash little r. They're both G positive. So this is where, again, it can get really confusing. So in the next 3 minutes, I'll try and explain this to you. So it's known what's removed and what's left behind. So if you do an absorption and you've got D plus C plus G, your R dash little r plasma removes the C and G, but would leave D behind because it's D negative. The D positive one removes D and G, but it leaves C behind. So I appreciate this is really confusing. So it's known what's removed and what's left behind. Therefore, your neat plasma has to contain all of them, and that is how we do it. And it's very confusing and it takes ages to get your head around. But by seeing what's removed and what's left behind, you can see if it's got all three there. If it's D and G, you would have your R dash plasma remove C and G, but it can't remove D, so the D's there. And the other one removes the D and the G, and there's nothing left behind, therefore it can't contain anti-C, because if the big C was there, it would be gone. So that's how we'll work out whether it's D and G without C. So therefore it's got to be D and G. And if you have C and G, then the C and G is removed. It doesn't contain anti-D because the D is not left behind. It's a D negative cell, so it would have to leave it. And this one here, it leaves behind the anti-big C. So you know, because you've removed, you're confirming that the G is there because you've removed it all out and it's left the big C behind. Therefore, it's got to contain C plus G. So, and when you're looking at your blood selection there, like, as I mentioned, you want to be going for your little r, because that's around 15% of your UK donor population. The other two phenotypes are really rare, so you probably wouldn't come across those. So sorry that was the quickest whistle stop tour ever. But if you do want some more educational resources, I appreciate these. This has been 3 sessions, and as I mentioned, each one of them could have been probably its own 3-hour-long session. But if you go over at the Transfusion Training Hub, there's loads of resources. There's links to talks, there's e-learning modules and all sorts. There's just a whole host of information there if you want to go and visit that. And I finished bang on time, but I'm available if anyone's got any questions before I want to nick off. And I do apologise for talking so much again.
That's okay, no worries. Thank you, Helen, for leading such an amazing series. It's been great for all levels of staff. So whether you're beginning or you are looking to refresh some of that foundational knowledge, I think, parts 1 and 2, and also this webinar as well helped to address that. I've received many messages from attendees who've attended all the sessions to say that they've really found this useful. So once again, thank you very much, Helen, for leading the series. And also thank you to everyone who joined us today. I'll be sending out certificates of attendance over the next few days. And if you attended as part of a group, please send me an email with the details of everyone else who attended in the group so that I can make sure everyone receives their certificate. And if you haven't done so already, I guess we don't have time for questions now, but if you haven't done so already, please do consider registering to the OnlyCells webinar mailing list. So whenever a webinar is organized, we can send you information via email. Once again, thank you very much for joining us, and I look forward to seeing you on the next one.
Thanks so much, everybody. I hope you've enjoyed it.